What you're about to see is a real virus, captured at 64,000 times magnification through real experiments, carefully brought into view, atom by atom. Invisible roads. This is Enterovirus D68, isolated during the 2014 outbreak in the United States. It has been linked to respiratory infections in children and, in rare cases, polio-like illness. The entire virus is built from 60 repeating subunits. Here is one subunit. Each subunit is assembled from 4 viral proteins: VP1, VP2, VP3, lastly VP4. And here is a ribbon diagram of the VPs. Ribbon diagrams are often used by scientists to visualize proteins more clearly, showing how the proteins are folded and organized. Together, these repeating proteins assemble into a remarkably ordered shell. The shell, or the capsid, follows an icosahedral geometry: 5-fold axis, 3-fold axis, 2-fold axis. Rotate the pattern around each axis, and you begin to see the same geometry reappears again and again. Proteins are not the only way to explore a viral capsid. Here is something visually exciting. The surface is colored by depths. Higher regions appear in warmer colors, while deeper regions shift toward blue and purple, almost like a microscopic landscape. Around the elevated plateaus, the surface drops inward, forming a circular trench known as the canyon. See? It really does resemble a canyon. Let's take a closer look using another strain. The canyon is also known as a receptor binding site. Once receptors bind, infection can begin. What you're seeing here is a recently discovered structure of EVD68 bound to part of its human receptor. Hard to see? How about this? Now you may see it better. Once attached, the capsid can begin to open. But how? Using viral protein coloring, we may now observe the capsid more clearly as it begins to loosen. This transitional state is known as the A particle. As the capsid destabilizes, proteins rearrange and small openings begin to emerge. This is an unstable form believed to prepare the viral RNA for release. But human body will respond like this. Here, antibodies bind around the canyon region, blocking interactions required for viral entry and interfering with infection. This is one of the principles behind antibody therapy, and the antibody you're seeing here is currently under clinical investigation. Enterovirus D68. Yet, this is only one of the invisible worlds. These viruses share a common architecture, yet create very different landscapes. Each tells a different story. This is only the beginning. Many invisible roads remain to be explored.